Good morning, all of you. So yesterday we started on with signal transduction or intracellular signaling, and we discussed of G proteins, which are a group of intracellular signaling proteins commonly used for sending a message inside the cell and processing of that message. There were two classes of G proteins that we talked about. What was the first class? Heterotrimeric G protein made up of alpha, beta and gamma, correct? So it was made up of three proteins and it was able to activate an enzyme in the membrane to carry out the signal, correct? What was that enzyme producing? You talked about two enzymes. The products that those enzymes were producing are called as what? Adenyl cyclase is the enzyme. What was the product that they were making? Cyclic AMP, correct? Or commonly, if I want to call them, they were called as second messengers, right? In a language that was understood by the cell. So one enzyme was adenyl cyclase, as she mentioned. What about the other enzyme that we talked? Phospholipase C, and what was its secondary messengers? IP3, DAG and calcium. Very good. And the other G protein that we talked of was RAS G protein, and RAS G protein is associated with one response. What is that one response? Gene expression. And RAS G protein, the receptor that was associated with RAS G protein is, there is something special about the receptor that are associated. They are catalytic receptors, correct? They had an enzyme in them. Now taking it further, RAS is not the only protein that is able to receive a signal from a catalytic receptor. Catalytic receptors, they also play a major role in carrying out signaling with the help of other proteins. So let's look at how catalytic receptor signaling is happening. Now catalytic receptors are called so because they have an enzyme present in them, and because of that enzyme they are called as enzymatic receptors. They are made by association of a single chain transmembrane protein binding with another single chain transmembrane protein and binding to the signal molecule or the ligand. So if this is your membrane, one single transmembrane protein connects with another single transmembrane protein, then binds to a ligand. This is your catalytic receptor, and it can bind to different signaling molecules. I have given you some examples here. These examples are not important for now. Okay, you just remember they bind to any ligand. Okay, and the catalytic receptor, because they have enzymes in their intracellular region, what is the name of that enzyme? Tyrosine what? Kinase enzyme. So all the catalytic receptors, they have the enzyme which is called as tyrosine kinase. It's a kinase, meaning what can it do? It can add a phosphate where? In the tyrosine, on the receptor, correct. So these catalytic receptors have tyrosine kinase activity, which is able to bring about phosphorylation of tyrosine, which are part of the receptor, correct. And once these tyrosines, they get phosphorylated, they become binding site for different adapter proteins to bind to them, and it is these adapter proteins that carry the signal further inside the cell. So when phosphotyrosine is there on the receptor, adapter protein will come and bind, and it is this adapter protein which will take the signal further into the cell, and ultimately a cellular response is achieved. Okay? However, in our cytosol, we have another enzyme which is called as tyrosine phosphatases enzyme. If it is a phosphatases enzyme, can you guess what it is doing? It removes a phosphate, correct. So in our cytosol, we have the enzyme tyrosine phosphatase, which actually remove the phosphate from the tyrosine in a very regulated or controlled manner in order to end the response to signals. So for example, catalytic receptor has phosphotyrosines. These phosphotyrosine binds to adapter proteins and a signal is sent in the cell. A cellular response has been achieved. Now once a cellular response has been achieved, the signal has to stop. Because earlier also I have mentioned, whether I was talking about cell cycle or I was talking about protein degradation, I said any cellular process that is happening, after a certain time period, it needs to stop once it is finished. It cannot keep on going, because if it will keep on going, that is abnormal for the cell and a disease can occur, right? Same with signal also. When a cellular response has been achieved, the signal has to stop now. So how to stop that signal? This tyrosine phosphatase enzyme, it will remove the phosphate from the tyrosines that are present on the receptor. So when there is no phosphate on tyrosine, no protein can bind to it and the signal transmission ends. Is it clear? Okay. So let's look at the structure of the catalytic receptors. Now I have given you a fair idea about what is the structure, but let's look at what are the common features that are shared by all the different types of catalytic receptors. So catalytic receptors, they are made by association of two or more single transmembrane protein chains. So this is one transmembrane protein and this is another transmembrane protein coming together to give you a catalytic receptor. They mostly two are present, but in some cases you will see more, maybe three. Okay? When the receptor gets formed, it has an extracellular domain. Why extracellular domain is needed? To bind to the ligand. So, yes, exactly. So, this is the extracellular domain. It is able to bind to the ligand. It has a transmembrane region that passes through the bilayer of the membrane, and when it is passing through the bilayer, it makes a spiral which is called as alpha helical. So as you can see, I have made spirals on it. So as it is passing through the transmembrane, it makes a spiral so that the protein can easily pass through the membrane. And then you have an intracellular or cytoplasmic region, which is this, and this is the region where the enzyme is present. Each protein that is involved in the formation of the receptor, each protein will have its own enzyme, and the enzyme is tyrosine kinase. Okay, it is tyrosine kinase. Clear? The structure is clear to everybody? Now let's look at the signaling mechanism. Okay. For signaling mechanism, what is mentioned here in the text is for you to go back home and read. I want you to focus on this picture. And in fact, if you understand this picture, your job is done. You don't even have to learn it. Okay? So please pay attention. I am going to repeat it twice. The very first step is the ligand comes into the environment of the cell where it needs to bind. So this yellow thing is the membrane of the cell, and these red squares that you see is the ligand. So number one step is that the ligand comes near the cell membrane. Now as soon as the ligand comes, two transmembrane proteins, they come close together. They come close but they don't bind. So the number two step is two transmembrane proteins, they come close. The number three step, when they come close, ligand binds. One ligand on one protein. Okay? And so it is mentioned here also. So this is 3 where ligand binding happens, and as soon as the ligand binding happens, receptor dimerization happens, meaning the receptor gets joined. So both of them are happening actually together. That is why I put 3 on both of them. So ligand binds and immediately the receptor gets dimerized. When I say dimerized, it means it is joined together now. And here I am saying it is dimerized because there are 2 proteins. If there are 3 proteins, you will call it as trimerization. Okay? So as soon as the proteins are dimerized or the receptor dimerization has happened, the tyrosine kinase that is present in the intracellular region, they become active. This is step 4. The tyrosine kinase becomes active. Now when the tyrosine kinase becomes active, now this is something that you should pay attention to. Do you see 2 arrows going cross? If you see this one going like this, and do you see the other one going cross? So what happens? The tyrosine kinase on the left chain will add a phosphate on the tyrosine present on the right chain. So cross. And the tyrosine kinase of the right chain will add a phosphate on a tyrosine on the left chain. So it is cross phosphorylation. Okay? And this cross phosphorylation is important for the tyrosine kinase to become active, to elevate its activity, so become more active. Okay? So what happens? The tyrosine kinase, once it is active, it does a cross phosphorylation on the opposite chains only on one tyrosine. So one phosphotyrosine on one chain and another phosphotyrosine on another chain. That's step 5. And in step 5, why step 5 is important? Because this enhances the activity of the enzyme. Now once this has happened, step 6 occurs, which is autophosphorylation on tyrosine. What does auto means? Self. So now when these tyrosine kinase are fully active, now they will phosphorylate tyrosines present on themselves. So now no cross phosphorylation, autophosphorylation. Tyrosine of the left chain will, tyrosine kinase of the left chain will add phosphate on tyrosines that are present on the left chain. Tyrosine kinase of the right chain will add phosphates on the tyrosine that are present on the right chain. So autophosphorylation. Okay. Now when these phosphotyrosines are there, they become binding sites for adapter proteins, because for the transmission of the signal you need adapter proteins. So the adapter proteins with their SH2 domain, they are able to bind to the phosphotyrosine. Okay? And once the adapter proteins bind, the signal gets transferred to other proteins. You will see, I have mentioned in the slide, that they can either bind, the adapter proteins can either bind using their SH2 domain or they can bind using their SH3 domain. Okay? No, there is no 4. There is only either 2 or 3. Okay? What are you saying? Step 4. I am going to repeat it again, so listen. So what's happening? Any cell where signaling needs to happen, first of all, the first step is the ligand comes in the vicinity of that cell. Correct? That's step number 1. As soon as the ligand comes close to the cell, 2 transmembrane proteins, they also come close together, but they don't join. That's the second step. The third step is each transmembrane protein binds to one ligand each, right? So ligand binding happens, and at the same time when the ligand binds, receptor dimerization is also happening. So both the actions are third step. The fourth step is the tyrosine kinase, that is, you will pay attention now, right? the tyrosine kinase that is present in each chain, it becomes active. And in the fifth step, the active tyrosine kinase will do a cross phosphorylation for one tyrosine. So this one will cross phosphorylate a tyrosine on the left hand side, and the left hand side tyrosine kinase will add a phosphate on the right hand side. Why? That's the fifth step. And why this is happening? Because this increases the activity of tyrosine kinase. Clear? After this, autophosphorylation on tyrosine start happening. Now once when the tyrosine kinase are fully active, they will start phosphorylating their own tyrosines. No cross phosphorylation anymore. So they will add tyrosine to their own chains. Now when there are many phosphotyrosines, they become binding sites for adapter proteins. Adapter proteins can bind to phosphotyrosine using their SH2 domain or SH3 domain. Clear? Okay. Now since we are talking about catalytic receptors need some proteins to carry out their signal further into the cell. And those proteins are called as adapter molecules, or you can call them as adapter proteins. Okay? What are these adapter proteins? They are actually a link or a chain between an activated receptor and between the other signaling molecules that are present in the cell. So if what you see here in blue is a catalytic receptor, so this adapter molecule or module is forming a binding with the receptor also, and it is also binding to the next signaling molecule. Okay? That is why it is called as an adapter. Clear to everybody? Yeah? Yeah. Adapter molecule is a link between the receptor and the other signal molecule. Okay? Okay, let's look at different adapter molecules. Let's talk about RAS again. So yesterday when I was mentioning to you about RAS, I had already told you that RAS can bind to a catalytic receptor by using an adapter molecule, or it can bind directly to RAS as well. Do you remember this? Now when RAS directly binds to a catalytic receptor, it itself becomes a adapter molecule. So let's take that condition. So for example, a ligand comes and the whole process happening, the proteins, they come close, then they bind to the ligand, then the proteins dimerize to give you a catalytic receptor. When they dimerize, the tyrosine kinase in the receptor becomes active, does a cross phosphorylation to further enhance its function, and then phosphotyrosines are, autophosphorylation happens, many phosphotyrosines are there. So that is happening, for example, on the catalytic receptor, and it has many phosphotyrosines. So here, since the adapter molecule is RAS, it will use its CH2 domain to go and bind to these phosphotyrosines. Now when RAS binds to these phosphotyrosines, the GDP that was present on the RAS, it gets converted, it gets replaced by GTP. So a RAS with a GTP is an active RAS, correct? This RAS will now activate another enzyme. I told it to you yesterday, it is called as RAF. So RAS will activate RAF, or the other name for that enzyme is MAPKKK or mitogen activating protein kinase. It's a serine protease. It is a serine protein kinase. That means it adds a phosphate to a serine molecule. So when RAF is activated or MAPKK is activated, it in turn activates another enzyme in the pathway, which is MAPKK. So one kinase less. Okay. Then MAPKK in turn adds a phosphate on the other enzyme, activating it, which is called as MAPK. So as you go down the pathway, the kinases are reducing. Earlier it was 3, it became 2, and then the third one that is getting activated is only one kinase. Okay. Now MAPK, the activated MAPK will enter the nucleus and it will activate the regulatory particles, which are transcriptional factors. When transcriptional factors in the nucleus are active, what does RAS do? So the transcriptional factor will bring about transcription of gene or gene expression. Okay, let's go through it once more. So for RAS, if it directly binds to a catalytic receptor, it itself is an adapter molecule. So when the catalytic receptor has many phosphotyrosines, RAS with its SH2 domain binds to these phosphotyrosine. Now in its inactive form it is attached with GDP, and as soon as it binds to the receptor, the GDP gets replaced by GTP. The active RAS will activate another protein called as RAF, or you call it as MAPKK. MAPKK will add a phosphate on the next enzyme, activating it. That next enzyme is MAPKK. MAPKK will then add a phosphate to the last enzyme, which is called as MAPK. MAPK will go into the nucleus. It will add or activate different transcriptional factors, and when those transcriptional factors are active, what happens? Gene expression. Okay. So RAS as an adapter molecule.
Here in this situation, the RAS is an adapter molecule.
Correct. Let's look at the other adapter molecule which is also involved in gene expression, and that is called as STATs. The full form of STAT is signal transductors and activators of transcription. That is why we call it as STAT. Okay. STAT is also, it has a SH2 domain, and it will bind to phosphorylated tyrosines that are present on the catalytic receptor. Remember, when we are talking about adapter proteins, the receptor is always catalytic receptor, and when it is a catalytic receptor, the enzyme always will be tyrosine kinase. Okay. So here also the catalytic receptor, when it is active, the tyrosine kinase will add phosphates on the tyrosine, giving you many phosphotyrosines. Now what happened? STAT is an adapter protein. It will come and bind to the phosphotyrosines, one on one chain, another on another chain of the receptor. Okay. Now when STAT is bound to these phosphotyrosines, the same tyrosine kinase enzyme of the receptor adds a phosphate on the STAT tyrosine as well. It is mentioned here. Tyrosine residue of the STAT are phosphorylated by the tyrosine kinase of the receptor. So the tyrosine kinase of the receptor adds a phosphate on the STAT, making it active. Now once STAT has been phosphorylated, they get detached from the receptor and they bind together to give you a dimer. It is mentioned here. Phosphorylated STAT, they combine together to give you a dimer of the STAT. When the dimer of the STAT gets formed, it moves into the nucleus, binds to specific gene, bringing about expression of certain genes. So STAT is also involved in gene expression. It is more or less similar to how RAS is working, but with some modification. So again, catalytic receptor is there. The phosphotyrosines on the catalytic receptor acts as binding sites for the STAT. So to each chain, one molecule of STAT will go and bind to phosphotyrosines. Now when STAT is bound, the tyrosine kinase of the receptor adds a phosphate on the tyrosine of the STAT also. Okay? When the STAT receives that phosphate, it becomes active, it detaches from the receptor. Both STAT, they combine together to give you a dimer, and this dimer of the STAT will then move into the nucleus, binds to the gene, and gene expression happens. Clear to everybody? Yes? And the last adapter molecule that is involved in insulin signaling. All of you know about insulin, right? What is it?
Molecules that carry glucose inside the cell.
They do not carry. Insulin does not carry the glucose, but it allows the movement of the glucose inside the cell. For insulin signaling as well, catalytic receptors are needed. So the receptors where insulin bind is also a catalytic receptor with tyrosine kinase activity. So the process remains the same that I have already mentioned in detail, the different steps. So here also when insulin comes, the chains for the insulin receptor, they come together, then the insulin binds to the extracellular binding domain and the chains they dimerize, and when the chains dimerize, the tyrosine kinase become active. It's the same process. When the tyrosine kinase becomes active, many tyrosines after cross phosphorylation, many tyrosines get phosphorylated on the receptor. Now the important thing or the thing that changes here is what is the adapter molecule. So in case of insulin, the adapter molecule is called as insulin receptor substrate or IRS. So for insulin to carry the message of insulin inside the cell, the adapter molecule is IRS. There are different types of IRS, IRS1 and IRS2. Most of the cells have them, they are widely expressed. Then you have IRS3 and IRS4. Metabolically or energy wise, 3 and 4 are very important because they are found on adipose tissue, on pancreas beta cell and on the liver. And IRS4 is the adapter protein that you will find in thymus, brain and kidney. Again, which IRS is found where, I am not going to ask you that. That is something more specialized which you will take up in year 3. Okay? So just remember, with insulin, it has a catalytic receptor with the same mechanism explained before. What is the adapter molecule? IRS. Now let's look at the picture. Same, it has a catalytic receptor. When insulin binds to it, the receptor gets dimerized. The enzyme is tyrosine kinase. Many phosphates, as you can see here in yellow, many tyrosines get phosphorylated. These phosphotyrosines are site for the binding of the adapter protein. What is the adapter protein? IRS, and here an example of one has been taken in the picture. So IRS binds. When IRS binds to phosphotyrosine, IRS also gets phosphorylated. You see, it is getting phosphate here and here and here. So similar to what is happening in STAT. In STAT also, the tyrosine present on it is getting phosphorylated. In IRS also, the tyrosines that are present on IRS are getting phosphorylated. Okay? Hold on. So when the tyrosine on IRS gets phosphorylated, IRS becomes active. And when IRS is active, it can transmit the signal to different signaling molecules depending on what action is needed. For example, IRS can send a signal to PI3 kinase enzyme. And when PI3 kinase enzyme receives the signal, its function is to send the GLUT transporters to the cell membrane so that glucose can come inside. You already know GLUT transporters, right? So one of the functions of PI3 kinase is that it sends the transporter to the membrane. So this means it is involved in glucose transport. Then it will also help in protein synthesis, glycogen synthesis and cell proliferation. Okay? Now if the active IRS sends the signal to STAT molecule, instead of sending it to PI3 kinase, it sends it to STAT molecule, then STAT molecule will bring about gene expression. It is mentioned here, activation of STAT affects gene expression. Yes, your question. Yeah. Yes, because it is a catalytic receptor, no? And I just mentioned that all the steps that are taking place in catalytic receptor that I have explained in the previous slide will apply here also, because it is a catalytic. Yeah, I cannot explain that slide on each slide, right? So you understand, if it is a catalytic receptor, all the steps that I have shown you before apply here also. Got it? Here what you need to know is the adapter molecule is different, and that adapter molecule can send signal to different proteins depending on what action needs to happen. Clear to everybody? Any question? Okay. Now yesterday I had mentioned that I would be doing an introduction on the next resource which is to be taken up tomorrow, because it's a long resource and I want to have a head start on it. So let me just give you a brief introduction on apoptosis. So kindly open your slides on apoptosis. Okay, so let's talk about what is apoptosis, or what in simpler words, cell death. Now the general nature of the cell is to stay alive in order to allow us to be alive, correct? However, there are certain circumstances or certain conditions in which the cell chooses to die, and that process where the cell chooses to die is called as apoptosis. And there might be many conditions for that. For example, a cell got infected with a virus. A virus enters into the cell. Now this cell does not want that virus to go to other cells, so the cell itself undergoes apoptosis, so the virus also gets killed. Okay, so that is by choice. Another condition is that we talked about in regulation of cell cycle, when the DNA damage cannot be repaired. So the body does not want a cell with a damaged DNA. That is when that cell chooses to undergo apoptosis. And there are other physiological conditions in which, in spite of not having any defect, still apoptosis happens. There is a whole slide on it. We'll talk about it tomorrow. But just remember that apoptosis is a normal physiological condition that is happening in all of us, maybe right now also. Maybe there might be some cells in your body undergoing apoptosis just to save rest of the cells. Okay? So, in this particular resource, I want you to understand what the process of apoptosis is, why it is happening, the mechanism, the components that are involved in the process, and can we compare apoptosis with other processes of death, cell death. So, we will try to compare apoptosis with necrosis. Have you heard of necrosis before? Can you give me an example for necrosis? Necrotic. So what was the meaning of that? Death of a, let's say if I put it crudely, of a body part. Correct? So all cells eventually die either by necrosis or by apoptosis. Now we talk about it. Okay? So necrosis is what? Necrosis is a passive pathological process, meaning when cells are undergoing necrosis, it is because of some defect or some disease, usually a disease or an injury. So it is induced by cell injury, and it happens for a group of cells. So necrosis does not happen for a single cell, it happens for a group of cells together. And when necrosis is happening, the cell membrane of the cell rupture. Rupture means it breaks. So the cell membrane, it breaks open, and all the content of the cell, it gets spilled out. Now when the content comes out, it affects the neighboring cells also, and the immune cells can, they get alerted that this cell has ruptured, we need to go to that area. And that is why that particular area that is undergoing necrosis, you will see inflammation in that area. That means there will be a lot of immune cells. Now normally when necrosis starts, there is, or not even, when initially necrosis start, it is not necrosis, it is just damage. Basically that damage is not getting repaired, that is why ultimately necrosis happens. So when initially that damage is happening and there is spilling, so immune cells come in that area and that area becomes inflamed, meaning that patient will have redness, that area will be hot, it will be painful because of immune cells present in that area. So whenever those cells are undergoing necrosis, inflammation happens in that area. Okay? On the other hand, apoptosis is an active non-pathological process. So usually when apoptosis is happening, the reason are not any tissue injury happening. The reasons are more non-pathological. And apoptosis is a normal physiological condition which is as important as cell division that happens for our cells. So this means if there is any disturbance in the apoptosis pathway, then it might lead to different diseases like cancer, like autoimmune diseases, like neurodegenerative diseases. Okay? So what happens in apoptosis? Apoptosis is a process that affects only individual cells. So it's not a group of cells, it is a single cell without affecting its neighboring cells. So neighboring cells do not get affected, only one single cell get affected, and it does not induce any inflammation. So when apoptosis is happening, neighboring cells do not know that apoptosis is happening, so there is no inflammation. Yes? See, when the breakdown of the cell membrane will happen, the contents that come out, they call in the immune cells. So the inflammation that is happening, as per se, if I specifically point out inflammation in any area where you are getting hurt is because of movement of the immune cells in that area. Okay? Now, one of the characteristics about apoptosis is that the cell membrane of the cell does not break, rather it becomes blebbed. Blebbed means that the cell membrane will become like this. It will have bulging like this. I have pictures which will show you blebbing, and in fact if you see it in 3D, the cell that is undergoing apoptosis has blebs which make it look like raspberry. All of you have seen raspberries, right? They have a bumpy exterior. In the same way, the cell also looks like a raspberry, with the cell that is undergoing apoptosis. Okay? Talking a little more about necrosis, just a slide or two, because the main resource is not about necrosis, it is more about apoptosis. So necrosis is what? It is a pathological process which is induced in our body after an acute injury or an acute disease that has affected a particular body part. And in necrosis, you know, is it a single cell or a group of cells? It's a group of cells that undergo necrosis at the site of injury. Wherever that injury has happened, it is not one cell, it is multiple cells that are undergoing necrosis. And in necrosis, the cell increase in volume, then they lyse. As a result, the intracellular content comes out, which is causing potentially damaging inflammation to the neighboring cells also. So if a cell, this is a cell that is undergoing necrosis, first of all it will become big in size, and then the cell membrane will rupture, the content comes out. Because that content comes out, immune cells come and they attack the neighboring cells also. That is why we say necrosis happens to a group of cell, not one single cell. Okay? And for necrosis to happen, several days are needed. Apoptosis happens in few hours. Necrosis happens in many days. A classical example of necrosis is gangrene. Have you heard of gangrene before? In context of? Yes? And no? Low temperature, exactly, frostbite. You wanted to say something? Because of gangrene, right? Good. So gangrene is a classical example of necrosis, and as your colleagues tell that gangrene happens to that area where, you know, in people who have long standing diabetes or people who are smoking for a very long time. Now in such people, again what I am telling you right now is just nice to know information, you don't have to memorize it. So gangrene happens in people who have uncontrolled diabetes for many years. Because of uncontrolled diabetes, the blood supply to their arms and to their feet, it becomes less. Now when it becomes less, the nerves also become weak, and now they no longer have much sensation in their foot. They cannot feel, what is happening on their feet. If you touch their feet, they will say, I do not feel much sensation. So such people, when they get hit on their foot, there is an injury, they don't feel the pain. The injury is there, and especially diabetic people, they are very obese. So if they have with, long standing diabetes, they do not take care of their foot also as much. So if they get an injury on the foot, it goes unnoticed because they don't feel the pain. And when already the blood supply is less, right? So the repair is not also happening for them. When the repair is not happening, the cells in the foot starts undergoing necrosis. As a result, slowly that body part becomes completely dead. And in order to save such patients, you need to amputate their or cut their body parts, right? You might see case studies coming your way in which people who have severe diabetes for a long time, they have to undergo amputation. Their finger, initially a small toe finger is dead, that gets amputated. Then slowly that necrosis is spreading to other areas also. So their foot needs to be amputated. So it's really sad when this happens, but yes, this is a condition that can happen, and it's an example of necrosis. Okay? Is it understood to everybody? I would like to stop here now. We will take it up tomorrow. Okay?